শিক্ষাৰ তৰফৰ পৰা আজি আমাৰ নতুন বছৰৰ আচলতে প্ৰথম অনুষ্ঠান আজি জানুৱাৰী 25 তাৰিখ আৰু সকলোকে গণতন্ত্ৰ দিৱসৰো অভিনন্দন জনাইছো শুভেচ্ছা জনাইছো আজি আমাৰ মাজত উপস্থিত আছে ডক্টৰ গৌতম বৰঠাকুৰ তেখেতক নিশ্চয় বহুতেই চিনি পায় তেখেত বৰ্তমান হিউষ্টনৰ MD Anderson কেঞ্চাৰ চেণ্টাৰত তেখেত ডিপাৰ্টমেণ্ট অফ লিউকেমিয়া যাক একচুৱেলি সহজ ভাষাত ক'বলৈ গ'লে ব্লাড কেঞ্চাৰ বুলি ক'ব পাৰে তেখেত তাত প্ৰফেচৰ হয় লিউকেমিয়া বিভাগৰ আৰু তেখেত এজন কৰ্কট ৰোগ বিশেষজ্ঞ অৰ্থাৎ আমি অংকলজিষ্ট বুলি ক'ব পাৰোঁ মেডিকেল টাৰ্মিনলজি ইউজ কৰিলে গৌতম দা অৰিজিনেলি শিৱসাগৰৰ তেখেতে আছাম মেডিকেল কলেজত MBBS কৰিলে আৰু তাৰ পাছত তেখেতে একচুৱেলি পেথলজি বিভাগত PGI চণ্ডীগড়ত MD কৰিলে আৰু পাছত তেওঁ পেথলজিত MD কৰাৰ পাছত তেখেত ইউনাইটেড ষ্টেটছত আহি তেখেতে মেডিচিনত MD কৰিলে ব্ৰুকলিনৰ পৰা আৰু তাৰ পাছত তেখেতে মেডিকেল অংকলজিত MD এণ্ডাৰছন কেঞ্চাৰ চেণ্টাৰত ফেলোশ্বিপ সম্পন্ন কৰিছে আৰু তেতিয়াৰ পৰা তেখেতে MD এণ্ডাৰছন কেঞ্চাৰ চেণ্টাৰতে আছে আৰু এতিয়া তেখেত তাতেই প্ৰফেচৰ হৈ আছে ডিপাৰ্টমেণ্ট অফ লিউকেমিয়াত আৰু তেখেত লিউকেমিয়াত বহুতো গৱেষণাত জড়িত হৈ আছে আৰু আপোনালোকে জানি সুখী হ'ব যে তেখেতে এতিয়ালৈকে অন্ততঃ 500 মান ৰিচাৰ্চ পেপাৰত তেখেত পাব্লিছ কৰিছে গতিকে তেখেত এজন অতি নাম থকা লিউকেমিয়া বিশেষজ্ঞ আৰু বিভিন্ন ধৰণৰ লিউকেমিয়া আছে আপোনালোকে জানিবই তেখেতে মাইলয়েড লিউকেমিয়া মাইলয়েডিছপ্লাষ্টিক চিনড্ৰম সেইকেইটা টাইপ অফ লিউকেমিয়াত তেখেতে বেছিকৈ সম্পৰ্ক আছে আমাৰ মাজত আন এজন লিউকেমিয়া বিশেষজ্ঞ তপন শইকীয়া দাও আছে আমি লাকি যে তপন দাও আমাৰ মাজত উপস্থিত আছে আমাৰ ভাল লাগিছে আৰু গৌতম দা আপোনালৈ আগবঢ়ালো মঞ্চটো আজি তেখেতে আমাক লিউকেমিয়াৰ বিষয়ে ক'ব তেওঁৰ আজিৰ টকটোৰ টাইটেল হৈছে এন আনফিনিশ্ড জাৰ্নি ইন ব্লাড কেঞ্চাৰ থেংক ইউ গৌতম দা অভাৰ টু ইউ
ধন্যবাদ ৰিফাত মানে সুযোগটো যে দিলা সেইটো কাৰণে বৰ ভাল পালো জানো যে তোমালোকে বহুত ভাল ভাল কাম কৰি আছা বৰ ভাল ভাল ডিছকাশ্যন হয় তোমালোকৰ ফৰামত বেলেঞ্চড ডিছকাশ্যন হয় চাইণ্টিফিক কালচাৰেল বহুত ধৰণৰ তোমালোকৰ ডিছকাশ্যন হয় গতিকে এই ফৰামটোত সুযোগটো দিয়াৰ কাৰণে ভাল পালোঁ মই কওঁতে মাজে মাজে ইংৰাজী অসমীয়া অলপ মিলাই ক'ম আৰু কাৰণ অলপ মাজতে অসমীয়াটো ক'ব পালে ভাল লাগে গতিকে গুড মৰ্ণিং গুড ইভিনিং হেৰি তাকে মোক মানে মই শিৱসাগৰ ডাঙৰ হোৱা মোৰ জন্ম শিৱসাগৰতে মই হাইস্কুল শিৱসাগৰতে পঢ়িছোঁ সেইকাৰণে শিৱসাগৰ চৰকাৰী উচ্চতৰ মাধ্যমিক বহুমুখী বিদ্যালয়তে মই পঢ়িলোঁ এটা কাৰণে মেনশ্যন কৰিছোঁ মানে তেতিয়াতো আমাৰ ক্লাছ চাইজবোৰ প্ৰায় 100-130টামান ল'ৰা থাকে তাৰ মাজতে আৰু আধাইটো বিশেষ পঢ়া শুনাত মনো নিদিয়ে তাৰ মাজতে আমাৰ মানে টিচাৰসকলে ইমান ভালকৈ পঢ়াইছিলে আৰু যদি কাৰোবাৰ পঢ়াৰ প্ৰতি অলপ মন আছে সেইকেইটাক অকণমান মানে জোৰ দিও পঢ়ায় সেইখিনি কৰি মানুহ কৰিলে তেওঁলোকে হয়তো নপঢ়োৱা হ'লে মানুহ নহ'লহেঁতেন সেইকাৰণে মানে আই ৰিয়েলি পে মাই ৰেচপেক্টছ তাৰপিছততো মেডিকেল পঢ়িছোঁ অসম মেডিকেল কলেজত তাতো সেই একেটাই কথা ক'ব খুজিছোঁ প্ৰি ক্লিনিকেল পেৰাক্লিনিকেল ক্লিনিকেল ডিপাৰ্টমেণ্টত বহুত খুব ভাল ভাল আমাৰ টিচাৰ আছিলে আৰু আমাৰ ক্ল'জ সম্পৰ্ক আছিলে সকলোৰে লগতে ঠিক মানে নিজৰ বহুতৰ লগতে ঘৰুৱা সম্পৰ্ক আছিলে যথেষ্ট কষ্ট কৰি পঢ়ালে মানে সেই বেচটো নোহোৱা হ'লে হয়তো বাকী পাছৰখিনি নহ'লহেঁতেন তাৰপিছত মই গ'লোঁ চণ্ডীগড় পঢ়িবলৈ PGI পঢ়িবলৈ তাতে পেথলজিত মই ৰেচিডেঞ্চি কৰিলোঁ পেথলজি কৰি থাকোঁতেই মোৰ সেই ব্লাড ৰিলেটেড পেথলজিত বেছি ইণ্টাৰেষ্ট হ'ল আৰু তেতিয়া মই মানে এটা ভাবিলোঁ যে মানে পেথলজি ডায়েগনচিছৰ ফালৰ পৰাটো কৰিলোঁ, তাৰ লগতে যদি ট্ৰিটমেণ্টটো কৰিব পাৰোঁ হয়তো ভাল আছিলে, কিন্তু সেই মানে সেই বেলেঞ্চ ঠিক নাপালোঁ ক'তো। সেই তেনেকুৱা ধৰণৰ বেলেঞ্চ এটা ধৰক ইংলেণ্ড বা অষ্ট্ৰেলিয়াত আছে, কিন্তু US-ত নাই। সেইকাৰণে মই US-ত ব্ৰুকলিন হস্পিটেলত ইণ্টাৰ্ণ মেডিচিনত ৰেচিডেঞ্চি কৰিলোঁ। তাৰ মাজতে প্ৰায় 4 বছৰমান সেই ভিছা সংক্ৰান্তৰ মানে দৰকাৰত মই গৈ পেলাই অলপ গাঁৱলীয়া মানে টেক্সাছৰ অলপ গাঁৱলীয়া জেগাত 4 বছৰমান ইণ্টাৰ্ণ মেডিচিনৰ প্ৰেক্টিছ কৰিবলগীয়া হ'ল। সেইটো এটা শিক্ষা আছিলে এপিনৰ পৰা, কাৰণ সৰু জেগা য'ত মানে কোনো স্পেচিয়েলিষ্ট নাই। এপিনৰ পৰা মানে ইট ৰিমাইণ্ডেড মি অফ মাই ফাদাৰ, কাৰণ মোৰ দেউতাও আছিলে শিৱসাগৰ ডাক্তৰ। তেতিয়া শিৱসাগৰতো স্পেচিয়েলিষ্ট নাই। দেউতাই বহুত সময়ত চাইকেল মাৰি পেচেণ্টৰ ঘৰত গৈ পেচেণ্ট চাই। গতিকে সেই মানে মোৰো হয়তো কিছু পৰিমাণ তেনেকুৱা ধৰণৰ সময় এটা গ'ল। তাৰপিছত মই ফেলোশ্বিপ কৰিলোঁ MD এণ্ডাৰছন কেঞ্চাৰ চেণ্টাৰত, আৰু মই থাকি গ'লোঁ। আৰু মোৰ বিশেষকৈ মোৰ যিহেতু ইণ্টাৰেষ্ট আছিলে ব্লাড ৰিলেটেড ডিজিজৰ ওপৰত, সেইকাৰণে মই ব্লাড কেঞ্চাৰতে স্পেচিয়েলাইজেশ্যন কৰি থাকি গ'লোঁ। আৰু তাকে মানে বেলেগ বেলেগ ধৰণৰ লুকেমিয়া আছে, কাৰণ মানে ব্ৰডলি যদি আমি ডিভাইড কৰোঁ, then you can divide it into acute or chronic. Acute-to there is an acute myeloblastic or myelogenous leukemia, then there is an acute lymphoblastic leukemia. Aru chronic leukemias can be divided into chronic myelogenous leukemia or chronic lymphocytic leukemia. Even within these classifications, now as we know more through genomics, mutation profile, etc., that even within these individual diseases, be it acute lymphoblastic leukemia or be it acute myelogenous leukemia, there are a lot of subtypes. So it's not like cancer is not one disease, right? Cancer is possibly, thousands of diseases. And similarly, we're realizing that even in AML or ALL, based on the mutation, chromosomal abnormalities, etc., all this can be divided into subtypes. And not only that biologically we can divide it into these subtypes, but also the treatment cannot potentially change, particularly in the space of the targeted tube treatment. So I'll talk a little bit later about that. So normally when we have development of the blood cells, we all have blood cells that are circulating in our blood all the time. And these blood cells are turning over very quickly. Some of them turn over a little slower, like the red cells, they turn over a little slower. But whereas the white cells, the granulocytes, etc., they turn over within a question of hours. So within a day, this turnover can be 5-6 times. You can have a turnover. So the original cell or the baby cell is the stem cell. And the stem cell at a very early stage divides into either a myeloid stem cell, which will give rise to white cells, red cells, and platelets. And on the other side, you have the lymphoid stem cell, which then from the stem cell, you have the development of the lymphoblast, and which is the, one of the baby steps. And then when they mature, they can mature into B lymphocytes, T lymphocytes, or the natural killer cells. And this is the component that we usually use for our body's immunity. Now, in leukemia, what happens is this maturation process gets arrested at some point. And if you get arrested in the myeloid blast state, then you can develop acute myeloid leukemia. If you have arrest in a lymphoid blast state, then you can have acute lymphoid leukemia. So in some way, we can think about leukemia or blood cancer as arrest of maturation of the normal process. Right? I'll talk a little bit more in that context. Now, every time somebody comes to us with a diagnosis of leukemia, their question is that what stage am I? Because in most cancers, like lung cancer, breast cancers, there is this standard staging system. Whereas in most leukemias, the staging system possibly does not apply because it's a cancer of the blood, and the blood is circulating all over your body all the time. So more for acute leukemia. In some cases, in a chronic lymphocytic leukemia, we have some staging systems. But for acute leukemias, there is truly no standard, no staging system really. And in general, for our convenience, we divide the diseases into, has it been treated before, like untreated acute leukemia? Has the leukemia gone into remission, or has the leukemia come back? So the cancer, as I was talking about, it's a multi-step process, right? It's not one event that leads to cancer. And this hallmarks of cancer was initially described about a decade back, and then subsequently this has been updated. As you can see, there are multiple hallmarks that a cancer cell acquire to become a cancer cell which is different than the normal cell. So one is all these cells, the cancer cells need to divide, they need to proliferate, right? So most of our body cells have these signals in us, but in cancer, that's a sustained, almost like a constant pressure of division. Then for the cells to divide, they will need metabolic input, right? They need to burn fuel to divide, right? So that system also gets deregulated in cancer. Most of the cells, most of the normal cells, they have a lifespan, right? The cells die as a process of their normal growth and development. Whereas in cancer, the cancer cells learn mechanisms to resist cell death. And now this we can potentially use as a vulnerability to treat cancers, particularly in leukemias. That's another example that I'll possibly bring down the line. So the cell death mechanism, the tricks that the cancer cell learn to not die, and that gives them an advantage over the normal cell. So there is a differential between the normal cell and the cancer cell, because the cancer cells know how to resist cell death. And the proteins that they use to resist cell death, we can target those proteins to kill them. So that's an example I'll bring. Then of course there's a lot of mutations. We you know about mutations in the DNA or loss of chromosomal material. So those are the hallmarks of cancer. And then other thing is that in the normal body we have systems of eliminating abnormal cells, and some of these cells can even be precancerous cells. Say, for example, our body is exposed to sunlight all the time, and sun can damage our skin, and the skin is exposed, right? But most of us do not develop skin cancers, and that's mostly because our immune system has a very efficient mechanism to take care of and to eliminate these precancerous cells. The cancerous cells also kind of learn mechanisms how to evade this immune-mediated surveillance. And then, one of the important things is that, as I talked about, that the cells, they divide, they mature, they grow. The cancer cells, they have an arrest of maturation. They divide, and then they don't die. And that what is called the replicative immortality. That means they can divide and still remain immortal for a long period of time. And so those are some of the hallmarks of cancer, and some of these also becomes vulnerabilities of cancer also, which we can use to treat. There's some other emerging hallmarks. One of the things is this phenotypic plasticity. So even if I say a patient has a lung cancer, and say the tumor there is one tumor sitting in the lung, which is 1 centimeter, there's another tumor sitting in the other lung, which is 2 centimeter, and say that the lung cancer has also spread to another place in the body, say into the liver, right? So if we sample each of these sites, there are differences between these cells. They're all lung cancer, right? But they have different mechanisms of survival. They have different mechanisms of dividing and remaining immortal. So that's the phenotypic plasticity. So the system kind of, even when we give chemotherapy, some of these cells escape, and that escape comes from this plasticity, so that they can shift from kind of one mechanism of survival to another mechanism of survival, and there can be hundreds of mechanisms of survival. So that's another kind of unique capability of the cancer cell. Whereas a normal mature cells, if you have a cell that is in your heart, that heart cell almost never can become another cell. So it's a committed cell, right? So that commitment does not necessarily exist in cancer. So the cancer cells can go back to their primitive state and can kind of shift from one to the other. And apart from mutations in the genes, there are other processes called epigenetic processes. Epigenetics means over and above the genetic processes. So the DNA and the proteins that are closely linked to the DNA, they can go changes very rapidly, right? And these changes are not because of mutations in the gene or mutations in the DNA. So these changes are more changes of addition of, say, a small molecule, say a phosphate molecule or acetyl molecule. These molecules, when they are added to the proteins that are linked to the DNA or to the DNA itself, the DNA behaves differently in different contexts, right? So even though there may be one mutation driving that cancer, but within that cancer, there could be areas where the cells behave differently, more based on these epigenetic changes. So kind of what I'm trying to, I guess, highlight is that when we say lung cancer, we also are possibly talking about 10 different diseases, if not 100 different diseases. And as the patient gets treated, as the disease becomes more and more refractory to treatment, more resistant to treatment, and then they acquire all these different hallmarks or different versatility. So this is the genetic versus epigenetic changes. In the genetic changes, you have this DNA nucleotides, right? Thymidine to adenine, AT, GC, guanine, cytosine. That's the pairing that happens in the DNA, right? And this is our kind of almost a basic biology that we know. Now, say if this T gets changed to, say, G, then there will be change on the other side because the G has to pair with C. So those are kind of substitution changes that can happen. Apart from substitution, you can also have pieces of DNA that can be inserted into the DNA, right? So basically, it can acquire additional DNA material, which is the insertion changes. Or you can have loss of DNA material, which is the deletion of DNA. So these are more of the genetic changes, and these genetic changes would come down to the daughter cell, right? Once the cell divides, some of the genetic mutations will be carried into the progeny of the cell. Whereas the epigenetic changes are much more mobile or much more flexible. So this is a methyl group that's being added to the cytosine in the DNA. And the DNA kind of is usually wrapped around these proteins called histones. And these histones have these tails, right? Lysine, these tails are mostly composed of an amino acid called lysine. And that lysine can be acetylated. Just think about this process that you have all these modifications of the DNA and the protein that is linked to the DNA. And based on what the current state of that, that particular part of the DNA is, the DNA can behave differently. And this is a very dynamic process. All these molecules can be added, moved, added again. So that's what adds to the dynamism. So compared to genetics changes, which are more static, the epigenetic changes are much, much more versatile. And that's what makes cancers more complex. And it's not only in cancer, these epigenetic changes also exist in the human body. Because if you just think, the cell in the heart and the cell in the skin, they have the same genetic material. right? That you have inherited from your parents. But why does a cell become a skin cell? Why does a cell become a heart cell? And many of that actually depends on the epigenetic changes, because the genetic code in each of these cells are the same. Okay, so how does acute leukemia develop? Since my interest in over a number of years have been in acute leukemias, particularly in acute myeloid leukemia. So leukemia is a cancer that starts in a tissue that forms blood. We talked about the stem cells in the bone marrow, and these are the stem cells. That's where most of our blood cells develop, our white cells, lymphocytes, platelets, red cells. In a person with leukemia, the disease stem cells, they do not mature in the right manner. So there is an usual hierarchy, right? You have the blast cells, then you have the progenitor cell, then it becomes the blast cell, and from the blast cell, you have all these mature cells that develop. So there is a very structured hierarchy in the bone marrow. But that hierarchy gets lost in cancer, and that's why the stem cells, they do not mature in the right manner. And that's what we call the maturation arrest. And this is one of the hallmarks of cancer. These abnormal cells are the leukemia cells. And unlike normal blood cells, the leukemia cells do not die when they should. So that's the resistance to death, which is another hallmark of cancer. Now, since these abnormal cells which are not dying, they are kind of sitting in the bone marrow, they're crowding out the bone marrow. So, and they're dividing there and not dying, right? So they will basically crowd out the normal component of the bone marrow. And that makes a situation where many of these leukemia patients, they actually present with low blood counts, right? We talked about different types of leukemia. As I said, that since I do more of AML or acute myelogenous leukemia, I'll talk a little bit more about acute myelogenous leukemia. I'll talk more about general concepts. I don't want to get into the nitty-gritty of treatment because that's something possibly you don't need to know. But I'll possibly share some concepts where the treatment is going and how we think about treatment of leukemia. So AML, it's mostly a disease of the elderly. There is a spike in the very early age between few months of life actually. But beyond that, in the children, mostly the AML is a relatively rarer entity. But around the age of 60 or so, there seems to be a spike in the incidence of AML. And then there is a male preponderance compared to females. And so you realize that most of our patients of AML are older patients. And because they are older patients, many of these patients may not be patients for intensive chemotherapy or for, say, bone marrow transplant, right? So that's where there has been this major focus of developing targeted therapies so that we can actually bring treatments to these patients, and whether we can cure them or not, and at least we can improve their survival. So the 5-year relative survival rate is about 33% or so at this point. What the relative survival is that many of us, normally মানুহৰ heart attack হয়, diabetes হয়, kidney failure হয়, সেইবোৰ কাৰণে we can die from there. This is more that is related to leukemia compared to the normal population or same age population. That's about 33%, which is not bad. And if you look at this curve, in 1975, this was less than 10% for AML. And now we are getting into the mid-30s or so. You can think about this as a glass half full or half empty. মানে আমি oncologist community অকমান আমি we're more hopeful, right? গতিকে আমি এইটো মানে glass half full হিচাপে ধৰো। কিন্তু particularly, some of us who have been, doing this for a while, we have seen this, steady trend in improvement, and that's what makes it ever hopeful for this. Across age group, and if you take all the ages over years, the purple of the MOM font is possibly the most recent trend. This is from a database that includes all the American population, US population. Over years, over decades, there have been improvement in survival. That's number one. Number two, as you can see here, over decades, particularly in the young population, the 0 to 14-year-old. Now, acute leukemia is not very common in this population, but their survival has been in the 70% or 80% range, which is, unheard of in the adult leukemia setting. As you go into this is the 15 to 40-year age group, it's still in the 60% range or so now. As you hit the 60s, now it drops to below 50. And now you come to the age 70 or so, the long-term survival, if in 4 years, you're down to possibly about 5%, 10% or so. And that's where there is some trend of improvement, but that trend has not been substantial. But hopefully, with some of the newer therapies, this will help. This will improve. When we get a patient of leukemia, there's a whole set of workup that we do before we make treatment decisions. And same is true for other cancers also, but for leukemia, due to the blood cancer. So we do a lot of this, blood testings, complete blood count. Then we do take a sample from the bone marrow, because bone marrow is where the leukemia develops, right? And then sometimes we, pull a sample, then we do a biopsy, and from pathologists like Rifat, then we ask them to do something called immunophenotyping. At immunophenotyping or PRI, we can differentiate between, say, acute lymphoid leukemia versus acute myeloid leukemia. So those kind of informations are very important at baseline for us to make a diagnosis. And then we do this chromosomal studies called cytogenetics, and then we look for different gene mutations or chromosome changes. This is more to refine treatment and also to have a sense that in the long run, how the patient will do. Like there are some mutations, like here I'm highlighting this mutation called NPM1. Patients who have this mutation, they tend to have a better outcome, right? Whereas if somebody has a mutation in, say, RUNX1 or P53, those patients are going to do substantially poorly. So those patients, we need to think from the very beginning that can I take this patient to a stem cell transplantation or a bone marrow transplantation? Then, of course, we have to take a family history, patient's medical history, because that part is important. Because if somebody has a kidney, a damaged kidney, or has a damaged heart, or has poorly controlled diabetes, those things we need to take into account when we decide on the treatment. Then, of course, other testing for infections, etc. In this, the one important thing is this geriatric assessment, where basically we look at, is the patient fit or frail? Because depending on if the patient is fit or frail, we may decide on what treatment we go for. Because even if I have a 70-year-old who is very fit, who is running a marathon, there we may decide that maybe that patient can be a candidate for intensive chemo. But on the other hand, I may have a 45-year-old who has a heart condition or a diabetes or a kidney condition. There I may say that maybe a gentler approach is a better approach. So it's not a one-size-fits-all kind of an approach. I mean, গোটেই totality to ল'ব লাগিব যে পেশেন্টৰ কি সিচুৱেশ্যন, ডিজিজৰ কি সিচুৱেশ্যন, তাৰপিছত সাধাৰণতে আমি ট্ৰিটমেণ্টটো প্লেন কৰোঁ. The general kind of goal is that we diagnose, and then the first step is remission. That means after the first or few rounds of treatment, has the leukemia kind of cleared out, right? Now, clearing out is a kind of difficult statement to make, but when we do a repeat bone marrow test and then we look for, say, leukemia cells under the microscope or in the flow cytometry machine, then we are trying to look that can we see even small amounts of leukemia there, right? And if that is not there, that's when we call it remission. But in reality, what we can detect is our limitation, right? Our diagnostic techniques, etc., may not be good enough to detect, say, 1 in a million cell. We may be able to do 1 in 10,000, but we may not be able to do 1 in a million cell. But if we leave that 1 in a million cell, tomorrow the disease is going to come back. So that's why we do this post-remission therapy, or what we also call consolidation chemotherapy, to try to eliminate that tiny amount of disease. Or in some cases, we take the patient to a stem cell or bone marrow transplant for a potential final goal of cure. Now, things to consider that when we start that initial chemotherapy, what we call the induction chemotherapy, there are disease factors that we need to take into account, the chromosomal changes, the mutations, because based on this, we can, to a certain extent, predict what the response is going to be. On the other hand, there are host factors also, right? And these host factors will determine whether the patient will die from the initial treatment or so, or do we need to modify our treatment so that the patient does not die from our initial treatment that we give. So a lot of things matter. Age matters, performance status matter, how fit the patient is, how much of disease is there, if there any other organ dysfunction, right? Liver, kidney, etc. And does the patient have some ongoing infections like pneumonia? As we talked about, because of the low blood counts, many patients with acute leukemia, particularly, their immune system is pretty much non-existent, and they're vulnerable to different infections. It could be pneumonia, it could be other bloodborne infections also. So we need to take the totality into picture before we decide what the right first chemotherapy is going to be. What determines outcome? Again, some of these are patient-specific, and these are mostly related to early death of the patient, which is, again, age, performance status, organ dysfunction, kidney, liver, heart dysfunctions. On the other hand, the disease specifics, and they mostly drive resistance to treatment. And these are, the information that we take into account are chromosomal changes, mutations. Did the patient have prior chemotherapy? Because many times acute leukemia, particularly AML, can happen in patients who have been treated, say, for breast cancer or lung cancer. Wherever a patient has seen chemotherapy before and now has developed leukemia, that leukemia is a much more difficult leukemia to treat. And that's why we need to take all that into account. And finally, this is an important concept that's kind of emerging now, what we call minimal residual disease, right? The minimal residual disease is what I was talking about. Is there any leukemia left behind? Right. Now, the testings initially, when I was training as a pathologist, perhaps we were looking at under the microscope. We take a bone marrow sample, put it under the microscope, and look. That can determine maybe one leukemia cell in 1,000, but that's not enough, right? Because we have billions and billions of cells of leukemia cells in the body, and if I can detect only one in 1,000, that's not good enough. Now, the techniques have changed. The flow cytometry that I kind of alluded to, that can possibly detect in 1 in 10,000, 1 in 100,000. And now there are a lot of these DNA-based testings are available, which we call next generation sequencing, where it's done with DNA or RNA sequencing. These techniques can go down to 1 in a million. So over the last, say, 20 years, the ability to detect even a small amount of disease has changed. And that has also changed for other cancers also. Similar tests exist for, say, colon cancer, lung cancer, etc. The stem cell transplants, as I said, that some of the patients, we know that we cannot cure by chemotherapy alone. Right? So those are the patients, very early on, we try to get them onto a stem cell transplantation. Most of you must have heard the term called bone marrow transplant. Stem cell transplant, bone marrow transplant is somewhat synonymous, means more or less the same thing, that আগতে যেতিয়া মানে stem cell transplantation কাৰণে collect কৰে donor the person who donates, most of the donation was from the bone marrow, right? So that's what, you put a needle into the bone marrow, suck the person's bone marrow out, part of the bone marrow out, and use those cells to, give back to the recipient, the leukemia patient. আজিকালি বেছিভাগে bone marrow biopsy নকৰাকে হয়, মানে ব্লাডৰ পৰাই ল'ব পাৰে. That's why we call them, now can we use the generic term, the stem cell, whether the stem cell is collected from the blood or the bone marrow does not quite matter in that sense. After we collect these cells from the donor, then the leukemia patient gets intensive chemotherapy, right? So that we try to wipe out every trace of leukemia that we can. Now, these chemotherapies are very strong, right? And if we give this chemotherapy and do nothing after that, the patient will die because the bone marrow will never recover, right? And so the blood counts will never recover. So the patient will remain kind of dependent on transfusions for their lives, and because of infections or other things, patient will die, right? The toxicity of the chemotherapy that is given prior to the stem cell transplantation, we try to kind of overcome that by giving back normal bone marrow cells or normal stem cells, in this case, from the donor. So that now the entire blood forming element in this recipient, the leukemia patient, now actually comes from the donor, right? And hopefully once the donor stem cells, they go to the patient's bone marrow, set up home there, and now that, mature in the normal process, because these are normal stem cells, right? So they know how to mature in the normal way. So once that happens, the patient's blood counts recover, right? So that's sort of the whole idea behind it. So in the early days of stem cell transplant or bone marrow transplant, the idea was that you hit the leukemia patient with very strong chemotherapy, almost like, sublethal kind of a chemotherapy, kind of almost take the person to the brink of death and revive the person by giving normal stem cells from the donor. And so that the normal, the blood counts in the recipient can recover. But over time, what we have realized is that that's not the only role that the stem cell transplant plays. Now, when we give the stem cells, as I had shown in the earlier slide, these stem cells from the bone marrow can also create not only white cells, red cells, or platelets, can also create lymphocytes. And the lymphocytes are the immune cells in our body. And these are the cells in a normal person, they are fighting, cancers or looking out for abnormal cells and eliminating abnormal cells all the time. They are circulating all over the body. They're doing this day in and day out, right? So now when we give this normal stem cells from the donor to the recipient, these normal stem cells also will give rise to lymphocytes in the recipient, right? And these lymphocytes will detect the leftover leukemia cells as foreign. And so they'll start a war against them. And that's what they will kill the leukemia cells. And since these lymphocytes, they live for a long period of time as memory cells. These memory cells, they have a memory that I had seen this enemy before, right? And if that enemy shows back up again, this lymphocyte can go there and kill the enemy. So that's the more important part of possibly the impact of stem cell transplant in leukemia. AML, as we talk, it's both a genetic and an epigenetic disease. The one mutation possibly does not make leukemia, at least acute leukemia in AML. You need multiple mutations, and these multiple mutations cooperate in different pathways. And basically, all these pathways kind of check the boxes of hallmark of cancer. It could be maturation arrest, it could be proliferation, it could be avoidance of cell death, it could be changes in the metabolism so that the cell can divide indefinitely and remain immortal. So these are the mutations for our convenience, we have been dividing them into class 1, class 2, sorry. Class 1 mutation, class 2 mutation, class 3 mutations, and possible class 4 mutations also. The class 1 mutations, they mostly allow these cancer cells to divide and proliferate, right? Where the class 2 mutations mostly are around maturation arrest. The epigenetic mutations that we talked about, that epigenetic process, they also mostly contribute to arrest in maturation. And then there's a class 4 mutations, mostly mutations in some of the pathways called p53 pathways. These basically make the cell, they evade cell death after seeing chemotherapy. So all the different hallmarks of cancer are driven by different sets of mutations in the leukemia. And you normally need a cooperation between multiple of these mutations for the development leukemia. So in a patient with leukemia, if we do a bone marrow and look for all these mutations, on the average, possibly each patient will have about 4 mutations. And as they get treated, and if the leukemia comes back, they can potentially acquire more and more mutations so that they can evade treatment and kind of remain immortal that way. So cytarabine or RSC is one of the standard chemotherapies that we use in the treatment of acute myeloid leukemia. And it's a nucleoside analog. So what it means is it resembles our body's normal DNA components. As we talked about adenine, cytosine, guanine. So cytarabine mostly resembles the cytosine, which is a nucleotide that we all have in our DNA, right? So this cytarabine, when we give it through the vein, it is taken up by the cell. And then this cytarabine kind of becomes phosphorylated, first by monophosphate, diphosphate, and finally triphosphate. And the triphosphate form of cytarabine is the one that gets incorporated into the DNA because these cancer cells are dividing, right? So they need, more and more DNA material to be synthesized so that they can divide, right? If they are not making more DNA, they cannot divide into 2 cells because you need a whole new set of DNA for the new cells that are coming from the parent cell. So into that DNA, the cytarabine gets incorporated, and now this is not a normal nucleoside, right? It's not a normal cytosine; it's an abnormal cytosine, and so then the cells cannot divide anymore, and the cell's division process becomes completely disrupted, and the cell dies, basically. So that's sort of the general concept of cytarabine chemotherapy. And some ways, the general concept of most of chemotherapy that is directed towards DNA synthesis. There are other ways of, damaging DNA in the cancer cells, but this is one way of doing it so that you provide the cancer cell with a nucleoside analog, which is somewhat similar to the normal nucleotide that the body uses to make DNA, and the cancer cell into incorporating this abnormal nucleotide into its DNA so that the cell will die. Now, you must have heard a lot about targeted therapies versus chemotherapy. The advantages of the targeted therapy is that the target cells that have mutations or a particular genetic change, the targeted therapies will only hit those cells, right? The target cells that rely on this particular pathway that is driven by this mutation, and they use these pathways to survive, right? And if you're killing only these cells that have these mutations or a particular pathway that the cell is using to survive, and if that pathway is different than a normal cell pathway, then you can avoid collateral damage, right? Whereas in chemotherapy, the chemotherapy does not differentiate between a normal cell and a cancer cell, because any cell that is dividing will possibly incorporate those chemotherapies into their DNA. And so you run a risk of hurting normal cells that frequently divide, right? Um, the limitations of the targeted therapies are that we are still learning about what to target in different cancers, what to target even in acute myeloid leukemia, what to target when they first come, what to target when the disease comes back, and new mutations or new pathways are acquired for the cell to survive. So some of these are more limitation on our side because we don't understand the biology enough. And then we talked about this plasticity that one cell, even within the AML patients, there can be very different types of AML cells within the same patient itself. And some of these cells will possibly die because of targeted therapies, and many of the cells will figure out a way to kind of survive these targeted therapies. And if there are multiple mutations, right, as I talked about, the average, we may have 4 mutations. As the patient becomes more resistant to treatment, that can go up to 6, 8, 10, 12. So if there are multiple mutations are driving the leukemia, if you target one mutation alone, that possibly is not going to be the answer. So in terms of targeted therapies, actually imatinib or Gleevec, that is possibly the poster child of targeted therapy. And that was coming up just around the time I was finishing my fellowship, actually. So Gleevec is used to treat patients with chronic leukemia. It's called a chronic myeloid leukemia. And this leukemia is driven by a particular set of genetic changes, which is called BCR-ABL translocation. So there is a piece from chromosome 9 and a piece from chromosome 22. They come together and now make a new protein, right? So this is the enzyme, or which is, the CML enzyme here, and which is actually the BCR-ABL enzyme. Now this enzyme binds ATP, right? And this ATP is taken as a donor of phosphate molecule, and that phosphate molecule is transferred by the CML enzyme into the cancer protein, right? What Gleevec does is basically competes with, it competes with the ATP, right? Instead of the ATP binding into this CML enzyme, the Gleevec comes and sits there so that the ATP cannot bind, right? So now this ATP cannot donate the phosphate molecule to the cancer protein. And so the cancer protein cannot do its job, basically making the cancer cell survive. And so that's how the CML cell will die. And this is what exactly happens, that the imatinib sits in this pocket called an ATP binding pocket, right? And it's like almost like a lock and key kind of mechanisms where you put the key into the lock and nothing else can come and bind there now. And so now, if there is no signaling, right, downstream of the cancer driving protein, the cancer cell will die. And this has been a kind of major success story in the field of chronic myeloid leukemia, that like 20, 25 years back, after about 4 to 5 years, the survival was about 5 to 10% only, right? So even though it is a chronic leukemia, after 4 years, the survival was about 5 to 10%. And now, with the current state of therapy, these patients can live a normal life, basically. Their lifespan, if they are responding well to the treatment, is similar to a normal person. So that's an amazing change by the use of targeted therapy. This has become more successful in the context of CML is because the CML is driven by this kind of chromosomal translocation, right? So the BCR-ABL is the driver for CML. So if you hit at the core of this, then you can actually cure CML. Whereas that's not possibly true for most of the acute leukemias because there are multiple drivers of disease, and that's why we don't see that level of success that we see in, say, CML. I'll give an example of a targeted therapy in acute myeloid leukemia. This mutation are in a genes called isocytosine dehydrogenase 1 and 2, or IDH1 and 2. This IDH, there are 3 IDH. One IDH is in a cytoplasm of the cell, and there are two IDHs in the mitochondria of the cell. And these IDH enzymes are very important for the Krebs cycle, which is the metabolic cycle that we all have in our body. And that's how, say, glucose and other things are utilized in our body and converted into energy so that we can survive. So this mutant, IDH2, it produces an abnormal metabolite called 2-hydroxyglutarate. Now, this 2-hydroxyglutarate can change the DNA methylation. So this methylation is the epigenetic change that we had been talking about. And this can basically leads to a block in the maturation of the cell. As we talked about, this block in the maturation of the cell is one of the hallmarks of cancer, right? Now, a drug has been developed that can potentially hit only the mutated IDH2. It does not hit the normal IDH2 enzyme. It only hits the mutated IDH2. So in that context, the patients with acute myeloid leukemia who have an IDH2 mutation, who are newly diagnosed, older patients who are not fit for intensive chemotherapy, right? As we're talking about patients in the age of 70, their long-term survival is what, 4%, 5%, something like that, without treatment, or even with treatment. Now, with some of the low-intensity chemotherapies called azacitidine, they about 50% of them would live about 8 months or so. Whereas when we added this inhibitor of this IDH2 mutation, as you can see in their kind of 50% survival has moved from about 8 months to now to about 2 years. So these are this is possibly not a cure, but this is definitely extending life. And most of these patients, they're getting treated not in the hospital, but as an outpatient. And this drug, which is ivosidenib, it's basically a pill. So just think about that older patients with acute leukemia, where it was a death sentence before. Now they're living for 2 years plus, thanks to some of these targeted agents. This is just an example. Now, this targeted example is of targeted therapy in the context where a particular mutation is present, right? Now I'm going to talk about another situation where there may not be a mutation, but without a mutation, the cancer cells still may be vulnerable to some of the other targeted agents. So normally, in the bodies, the cells are growing, cells are dying. So we have this intrinsic cell death pathway that is active in our cells, right? And in the body, there is a very fine balance between this tendency for the cell to die and the tendency for the cell to survive. And there are two pathways of cell death. I mean, there are multiple pathways of cell death, but there is one pathway called apoptosis. And this apoptosis can be driven by extrinsic pathway or by the intrinsic pathway. The intrinsic pathway is driven by mitochondria. What happens when the intrinsic pathway of death is triggered? Say the cell is exposed to some damage, or say the cell has seen chemotherapy, or the cell has seen some sort of a damage, and the cell just cannot recover anymore, and the cell will die. And in that process, there are enzymes that are released from the mitochondria, which are called cytochrome C. And this cytochrome C in turn activates some of these enzymes called caspase enzymes. The caspase enzymes are in our cells, but they remain inactive. But whenever the cytochrome C is released from the mitochondria, as you see, the cytochrome C is kind of sequestered in a mitochondria. So in a normal cell, the cytochrome C is not floating around here, right? It is sequestered, whereas the enzymes that are executioners of death, they're in the cytoplasm, right? So the cell is keeping them separate. Right? Now, if in the process of the cell dying, if the cytochrome comes out of the mitochondria into the cytoplasm, now these executioner enzymes get activated and they'll trigger cell death. So in that process of so-called apoptotic cell death or apoptosis, there are proteins that will resist apoptosis called the anti-apoptotic proteins, and then there are cells that are pro-apoptotic. They will drive cell death, and that's the balance that kind of maintains the fate of the cell, whether the cell dies or the cell lives, right? Now, in the leukemia cell, of course, the balance is tilted to survival because they're trying to become immortal. But this survival balance is not tilted by any mutation, but by increased expression of some of these anti-apoptotic proteins, anti-cell death proteins, right? So now, since this cancer cell is dependent on these proteins to survive, now can we target them, and can that be a vulnerability in the cancer cell? So that's what has happened. A drug that has been developed called venetoclax, and that basically binds to this anti-apoptotic protein, which is in the green, called BCL2. What the BCL2 does, the BCL2 kind of binds to the proteins that are more pro-death, right? That can drive death, and they kind of block these proteins from causing cell death, right? That causes the cancer cell to survive, because in this balance, because the anti-cell death protein is stopping the pro-cell death protein, the cell survive. Now, if we can put in a small molecule, which, like as I was talking about in the context of CML, that can bind to this BCL2 protein, and that will release the pro-cell death protein, and that will eventually cause cell death by release of the cytochrome from the mitochondria, right? And that's what has been used in the clinic. And this is for a patient population which are over the age of 75. So again, as we talked about, these patients had a survival rate in a year of about 3 to 5%. And now in this patient population, when we used the venetoclax, which is the drug that drives cell death, and add it to low-dose chemotherapy, versus we compare low-dose chemotherapy, with basically a sugar pill, there is a definitely higher rate of response, right? And these patients also survive better, right? Their average survival otherwise with low-dose chemotherapy would have been about 10 months. Now you get another 5 to 6 months. And we are kind of working on this to see if we can add other targeted agents to this backbone of low-dose chemo plus venetoclax, add a third agent to see if we can improve upon that. And that's happening. And that's the work in progress at this point. Now, finally, all of you must have heard about immunotherapies in cancer, and that's an exciting field because you are not using chemotherapy, right? That way you can potentially avoid some of the chemotherapy-related damages to normal tissue. So how do you activate an immune system? We all have our immune system in our body, right? This immune system is trying to fight infections, trying to fight cancers, trying to kind of keep us safe, basically, right? But the immune system, if it gets too ramped up, right, then you can have autoimmune diseases, right, where the immune system can turn against yourself, right? You must have heard about conditions called lupus, rheumatoid arthritis. Those are autoimmune processes where our own immune system is trying to basically, fight our own body, right? Now you can use the immune system to fight cancer. How do you activate the immune system to fight cancer? Now just think about driving a car, right? How does the car move? You can take the brakes off, right? Or you can press on the accelerator. And taking the brakes off is the normal immune system. It kind of requires it to be dialed up and down. And there are molecules called checkpoint proteins which can dial it down. Now, there are antibodies or drugs that have been developed where you can block these checkpoints, right? And cancer uses the same mechanism, uses these checkpoint proteins to dial down the body's immune system so that the body's it can remain invisible to the immune system, basically, right? But what you can do, if you can block this checkpoint protein, that it will possibly take the brake off, and then your immune system can potentially fight cancer cells. In some cancers, it's been very successful. lung cancer, some of the skin cancers called melanoma, kidney cancers, colon cancer, multiple areas where they have been very, very successful. And that actually has led to a Nobel Prize that was awarded to Jim Ellison several years back. And interesting, actually, Jim Ellison, he's a faculty member in our center. And one of younger colleagues from Assam, Sangeeta Goswami, she actually works very closely with Jim Ellison. At some point, hopefully you guys can have Sangeeta in your forum where she can talk about this much better, because she's much more qualified than I am to talk about that. So similarly, you can also press on the accelerator. You can use antibody drug conjugates. You can use antibodies to deliver chemotherapy only to the cancer cells, because if you have antibodies that can recognize a certain proteins on the surface of the cancer cell and not on normal cells, and then you can tag these antibodies with chemotherapy, and so the very targeted delivery of chemotherapy, right? Then there are other kind of mechanisms of activating the immune systems called T-cell engagers. And many of you might have heard about these CAR T cells. I'll talk a little bit about CAR T cells just to give an idea about what it is, because I'm pretty sure that many of us will hear about this over and over again in the years to come. So talk about this normal T cell, which is a lymphocyte, right? This normal T cell has receptors on the surface, which is called T cell receptor. And in the tumor cell, some of the proteins in the tumor cell that are processed inside the cell, and they are presented on the surface of the tumor cell, right? And those proteins can be recognized by the T cell receptor of the normal T cell. And then this T cell will try to kill the tumor cell, right? As you said, that some of these processes can be blocked by the checkpoint proteins so that the tumor cell remains invisible to the normal T cell. What you do is, in the CAR T cell, the CAR T cell is a chimeric antigen receptor T cell, right? What is a chimera? The chimera actually comes from Greek mythology, where a chimera is a fusion of multiple animals. The chimera has a head of a lion and another second head of a goat and a tail of a snake. So this is the mythological chimera from Greek mythology. Now, what is done in the CAR T cell context, you remove the normal T cell receptor from the T cell, right? And you put in new kind of molecules onto the surface of the T cell, which is a chimeric protein. So the chimeric protein takes a piece of an antibody that can detect proteins on the surface of the cancer cell. So these are targets that are expressed on the cancer cell and not on the normal cell. And then you add a piece of signaling domain so that when this interaction happens, this signaling domain activates the T cell. Basically hyperactivates the T cell. And then this T cell becomes a T cell that can fight and kill the tumor cell that carries this particular protein on the surface. So very targeted killing of the cell. So that's a concept that is becoming fairly active in some leukemias, particularly in acute lymphoblastic leukemia. This has been very successful. And the CAR T cell products are also available for acute lymphoblastic leukemia in India. And I believe the results have been very promising. Tapan Sarkar sir, hopefully Tapan can talk a little bit more about that. But this is sort of the next frontier we're trying to overcome in the space of acute myeloid leukemia, also that you can use the T cells from the patient, right, and make CAR T cells in the lab, so that you arm these T cells with these particular tools, the chimeric tools, and add the chimeric antigen receptor now, and grow the cells in the lab, outside the body, and into millions of these cells. From a few thousands, you can make millions of these cells and put these millions of cells back into the patient, so that these CAR T cells can look for these cancer cells or the leukemia cells and then kill the leukemia cells very specifically. Yeah. So that's sort of the holy grail that we are all looking for. And thanks for listening. This is a sunrise in actually 2 continents. Number of years back, we traveled to Nepal, and I saw the sunrise in one of the peaks in Nepal. And a year back, we traveled to Argentina, and we saw almost a very similar picture. So this is the tip of Argentina, and this is in Nepal. So sunrise in 2 continents. And again, my email is at the bottom. Please free to contact anything that I can answer, and I'll be more than happy. Thank you.
Thank you so much, Gautam da, for this very detailed overview, and still at such a short time on so many different aspects of leukemia. Thanks a lot. Before we go into the Q&A session, I just want to have a very quick announcement from our side. Just give me a minute. So I want to let the listeners know about our next lecture. আমাৰ এটা Nobel প্ৰাইজৰ ওপৰত এটা চিৰিজ চলি আছে, চ' গতিকে মানে এইটো সেইটোৰ লগত সংগতি ৰাখি ফেব্ৰুৱাৰী 8 তাৰিখে এটা লেকচাৰ হ'ব ইকনমিক্সৰ 2025 Nobel প্ৰাইজৰ ওপৰত, এণ্ড আমাৰ স্পীকাৰ হ'ব হিৰণ্য দা, ডক্টৰ হিৰণ্য নাথ, তেখেত ছেম হিউষ্টন ইউনিভাৰ্চিটিৰ প্ৰফেচাৰ ইকনমিক্সৰ। আৰু এইখিনিতে আপোনালোকক জনাবলৈ বেয়া সুখী হৈছে যে আমাৰ অনুষ্ঠানটো 15 মিনিট শিক্ষাই পঞ্চম বৰ্ষত উপনীত হ'ব, 5 বছৰ পূৰ হ'ব এই বছৰ। আৰু সেই উপলক্ষে আমাৰ এটা 5th ফাউণ্ডেশ্যন ডে লেকচাৰ হ'ব, আৰু সেই লেকচাৰটো হ'ব ফেব্ৰুৱাৰী 22 তাৰিখে। আৰু বহুতে হয়তো চিনি পায় অনুপম দত্ত, চ' তেখেত অসমৰে হয়, তেখেত কটনৰ আগৰ ষ্টেটিষ্টিক্স ডিপাৰ্টমেণ্টৰ প্ৰফেচাৰ জিষ্ণু দত্ত ছাৰৰ পুত্ৰ। চ' তেখেত বৰ্তমান প্ৰিন্সিপাল ৰিচাৰ্চ চাইণ্টিষ্ট হয় স্ন'ফ্লেক নামৰ এটা কোম্পানীত, আৰু তেখেত ষ্টেনফৰ্ড ইউনিভাৰ্চিটিৰো এডজাংক্ট ফেকাল্টি। চ' হি ইজ ভেৰি প্ৰমিনেণ্ট ইন দ্য ফিল্ড অফ AI। চ' তেখেতে 5th ফাউণ্ডেশ্যন ডে লেকচাৰ দিব, আৰু আশা কৰোঁ আপোনালোকে এটেণ্ড কৰিব পাৰিব। চ' এগেইন বেক টু দ্য মিটিং। আৰু কাৰোবাৰ যদি কিবা প্ৰশ্ন আছে সুধিব পাৰে। এটা প্ৰশ্ন একচুৱেলি মই ইউটিউবত দেখিছোঁ, মই মেবি মানে এইটো কৈ দিওঁ পটককৈ, যে এটা প্ৰশ্ন আহিছে মানস শৰ্মাৰ পৰা, যে কেন এপিজেনেটিক মেকানিজমছ ছাপ্ৰেছ কেঞ্চাৰ গ্ৰোথ? And a related question is, so epigenetic changes can impact mainly on gene expression? আপনি উত্তৰ দিব পাৰিব গৌতম দা, প্লিজ?
ঠিকেই প্ৰশ্নটো, কাৰণ এপিজেনেটিক প্ৰচেছটো বেচিকেলি জিন এক্সপ্ৰেছনটো কণ্ট্ৰল কৰে, ৰাইট? যেনেকৈ ইমিউন চিষ্টেম ডায়েলিং আপ ডায়েলিং ডাউনৰ কথা ক'লোঁ, তেনেকৈ কিছুমান আমাৰ জিন ধৰা বেলেগ বেলেগ সময়ত চেলটোক বেলেগ বেলেগ প্ৰটিন দৰকাৰ হয়, গতিকে সেই প্ৰটিনবোৰ বেলেগ বেলেগ সময়ত কেনেকৈ মানে চেলটোৱে synthesize কৰিব পাৰে। গতিকে সেই the genes that drive this protein expression, right, they are turned on and off, and this turning on and off is again based on epigenetics. Exactly. Because if all the genes are turned on all the time, then either the cell will not survive, or the cell cannot go through the normal maturation process. All right. And sometimes the other example that people give, that say sometimes even twins may have very similar, they basically have exactly the same DNA content, right? But sometimes they may look different too, and some of the differences or their development, etc., can also be because of the epigenetic changes. So even with the same genetic content, right, the epigenetic is what is basically driving the expression of a gene or turning off of a gene. So if you have a gene, say that is anti-cancer, right, or what we call tumor suppressor genes, right? So those genes can be turned on by epigenetics, right, and then you can potentially target cancer by that. On the other hand, the cancer figures out a way how to dial down this tumor suppressor genes, right? And on the other hand, the cancer figures out how to turn on the oncogenes or the cancer driving genes. So these are mostly epigenetic driven. That's correct, and that is another area of therapy that is being developed also.
Thank you, Gautam da. So actually এইখিনিতে মানে মই আজি আমাৰ মাজত আমাৰ সৌভাগ্য যে তপন শইকীয়া ছাৰো উপস্থিত আছে। তেখেত নিজে এজন অতি known renowned leukemia expert. তপন দা আপোনাৰ যদি মানে অলপ কমেন্ট দিয়ে এই টপিকটোৰ ওপৰত গৌতম দাৰ লেকচাৰৰ ওপৰত আৰু আপোনাৰ ফালৰ পৰা প্লিজ we will be
ধন্যবাদ ৰিফাত। গৌতমৰ কথাখিনি অতি মন দি শুনিলোঁ। ভুৰুকাত হাতী ঘূৰোৱা সহজ নহয়। কেঞ্চাৰ অতি জটিল ৰোগ, তাৰোপৰি তেজৰ কেঞ্চাৰটো আৰু কঠিন। আমি দৈনন্দিন নতুন নতুন কথা জানিবলৈ পাইছোঁ কেঞ্চাৰৰ বিষয়ে, বিশেষকৈ তেজৰ কেঞ্চাৰৰ বিষয়ে। খুব ধুনীয়াকৈ কৈছে গৌতমে। শ্ৰোতাসকলে ক'ব কিমান বুজিব পাৰিলে, কাৰণ বিষয়বিলাক ইমান কঠিন। হায়াৰ লেভেলৰ বিজ্ঞানৰ ষ্টুডেণ্টে কথাবিলাক বুজিব পাৰিব, এটলিষ্ট কথাবোৰৰ ভিতৰলৈ সোমাই পেলাই বুজিব। গোটেইখিনি কথা মোৰ কাৰণে সহজ, কাৰণ দৈনন্দিন জীৱনত সদায় ব্যৱহাৰ কৰি আছোঁ। কিন্তু গোটেইখিনি কথা যদি ভিডিঅ'টো কোনোবাই বাৰে বাৰে ঘূৰাই ঘূৰাই চাই বহুতখিনি কথা জানিব পাৰিব। চ' ধুনীয়াকৈ কৈছে গৌতমে। অসমীয়ালৈ অনুবাদ যদি হয় ভাল হ'ব, কাৰণ গৌতমৰ কাৰণে সহজ নহয় অসমীয়াত ক'বলৈ, কাৰণ 24/7 গৌতমে ইংৰাজীতে এইবোৰ কথা ভাবে, কয়, কৰে। আমাৰ কাৰণে ভবাটো অসমীয়াত ইংলিছলৈ ট্ৰান্সপ্লান্ট কৰা, কিন্তু এইটো নহয়গৈ, বুজিছা। আমি এতিয়া থাকোঁতে নহয় আমাৰ থট প্ৰচেছটো ইংলিছ হৈ গ'ল। অসমীয়াৰ পৰা ইংলিছলৈ নাযায়, ইংলিছৰ পৰা ইংলিছতে থাকে, কিন্তু ইংৰাজীৰ পৰা আমি অসমীয়ালৈ অনুবাদ কৰিবলৈ চেষ্টা কৰোঁ। মইতো সদায় কৈ থাকিব লাগে সেইকাৰণে। চ' সেইকাৰণে এটা সময়ত মানে মানুহৰ হেৰি মস্তিষ্কটো মানে বেলেগ ধৰণে আহিছে ইম্প্ৰেশ্যনটো। তুমি কিন্তু ধুনীয়াকৈ কৈছা যিখিনি কৈছা। কেইবাবাৰো চালে যদি য়াং ল'ৰা ছোৱালীবিলাকে বহুতখিনি কথা জানিব, কাৰণ তুমি পিছৰ কথাবিলাক ক'লা, এতিয়াৰ কথা ক'লা, ভৱিষ্যতত কি হ'ব পাৰে সেইখিনিও ক'লা। বৰ ধুনীয়া হৈছে, ভাল লাগিল।
ধন্যবাদ তপন দা। আপোনাৰ পৰা এইটো শুনি আৰু that makes a lot of difference.
ধন্যবাদ তপন দা। আৰু আজিৰ অনুষ্ঠানত বহুতো চিকিৎসক বন্ধু উপস্থিত আছে আৰু আন বহুতো উপস্থিত আছে। আপোনালোকৰ যদি কাৰোবাৰ কিবা প্ৰশ্ন আছে প্লিজ ক'ব পাৰে, আৰু আপোনালোকে আপোনাৰ হেণ্ড ৰেইজ কৰিলে আনমিউট কৰিও নিজেই সুধিব পাৰিব বা টেক্সট কৰিব পাৰে প্লিজ। গৌতম দাৰ দাদাও আছে ইয়াতে আজি, উত্তম দা। চ' মেবি উত্তম দাৰ কিবা প্ৰশ্ন থাকিব পাৰে। উত্তম দা?
নাই, একো প্ৰশ্ন নাই।
আপোনাকো লগ পাই ভাল লাগিলে। আপোনাৰ কথা শুনি আছো, লগ পোৱা নাই কেতিয়াও।
মই আপোনাৰ জানো আপোনাৰ বিষয়ে।
নাই, তুমি বুলি ক'ব। আৰু আমাৰ ভিতৰৰ সংঘমিত্ৰা হিৰণ্য দা, আপোনালোকৰ কিবা প্ৰশ্ন থাকিব পাৰে?
নাই, খুব ভাল লাগিলে, মানে বহুত কথা শিকিলো। মই মানে এইটো কথা নাজানো, কিন্তু এজ এ জেনেৰেল মানে বহুত কথা শিকিলো। ধন্যবাদ গৌতম দা।
হেৰি আমাৰ মাজত মোৰ বন্ধু কেইজনো আছে। মই হেমন্তৰ কথা কৈছিলো। হেমন্ত, তই আনমিউট কৰিব পাৰিবি নি?
মই আনমিউট কৰিবলৈ কৈছো। হেমন্ত দা আপুনি আনমিউট কৰিব পাৰে প্লিজ। হেমন্ত দা, আপুনি আছেনে? হেমন্ত দা?
আছো।
তাকে হেমন্ত, মই তাকে ৰিফাতক কৈছিলো যে আমি ক্লাছ 4ৰ পৰা একেলগে পঢ়া। তাকে আৰু হেমন্ত, এটা কাৰণে মই হেমন্তৰ নামটো ললো কাৰণ হেমন্ত পিডিয়াট্ৰিক চাৰ্জাৰী প্ৰফেচৰ অসম মেডিকেল কলেজত মেডিকেল কলেজখনৰ কাৰণে হেমন্তে বহুত কৰিছে আৰু মানে অসমত অন্তত পিডিয়াট্ৰিক চাৰ্জাৰী গ্ৰ' কৰাৰ ক্ষেত্ৰতো বহুত কৰিছে আৰু একাডেমিক পাব্লিচিঙৰ ক্ষেত্ৰতো হেমন্ত যথেষ্ট এক্টিভ গতিকে মই হেমন্ত তই যদি এবাৰ এইটো ফৰামত প্ৰেজেণ্ট কৰ বৰ ভাল পাম
হেমন্ত দা
মই আজি
নিশ্চয়
মই আহিলো মই প্ৰথমবাৰৰ কাৰণে আজি এটেণ্ড কৰিলো সেইটোৰ পোষ্টটো পাই পেলাই মই ভাল লাগিলে গোটেইখিনি কথা তমন নাই কোৱাৰ নিচিনা যথেষ্ট এটা ডিফিকাল্ট টপিক তই ইমান ধুনীয়াকে কলি তাকে মানে মই ভাবো যে আমাৰ যিখিনি অডিয়েঞ্চ আছিলে তেখেতসকলৰ কিবা যদি খোদনিও আছে ডেফিনেটলি তেখেতসকলে ক্লিয়াৰ কৰিব কিন্তু আমি ইভেন ফৰ দ্য স্পেচিয়েলিষ্ট অলছ' এইটো যথেষ্ট হেল্পফুল গতিকে এনেধৰণে আগলৈ হৈ থাকিলে ভাল পাম আৰু মই দিয়াটো বাৰু চাই ল'ম মই কথা পাতি ল'ম কেনেকে দিব পাৰি
আপোনাক কণ্টেক্ট কৰিম হেমন্ত দা ডেফিনেটলি
নাই ঠিক আছে অলৱেজ ৱেলকাম মই
হেমন্ত তই জান চাগে ৰিফাত AMC পৰা পাছ কৰা
মই আপোনাৰ ষ্টুডেণ্ট
ৰিফাত
আছিলো
জানো ৰিফাতক জানো আমাৰ AMC আজিকালি ল'ৰা ছোৱালী বহুতখিনি বাহিৰত যথেষ্ট ভাল কাম কৰি আছে ভাল কৰি আছে যথেষ্ট ভাল কৰিছে এইখিনি আমি ভাল পাওঁ সেইকাৰণে শুনিলে আৰু সিহঁতেও কেতিয়াবা AMCৰ কথাটো সদায় উনুকিয়াই থাকে আৰু ভাল লাগে সেইটো যে আমাৰ পেৰেণ্ট ইনষ্টিটিউচনৰ বিষয়ে এষাৰ কথা ক'লে আমাৰ সকলোৰে বুকুবিলাক ফুলি আহে সেইটো ভাল কথা আৰু ভাল কাম কৰক সকলোৱে আৰু হাউ উই কেন কণ্ট্ৰিবিউট আমাৰ নিজৰ স্পিয়াৰত সেইটো ডাঙৰ কথা আৰু গৌতমৰ কাৰণেতো আমি সদায় গৌৰৱবোধ কৰোৱেই গতিকে আজি এই ফৰামত লগ পাইতো সকলোকে লগ পাই ভাল লাগিলে শান্তনু দাকো দেখিছো মিতালী আছে আৰু যথেষ্ট
আৰু অন্তত মানে মোৰ বেটছটো লৈও মই অলপমান গৌৰৱবোধ কৰো কাৰণ আমাৰ বেটছৰ ল'ৰা ছোৱালীখিনিয়ে যথেষ্ট ভাল কৰি আছে মানে আই এম ৰিয়েলি প্ৰাউড নহয় মানে অসমৰ বেলেগ জেগাত আছে বহুত ভাল কৰি আছে তাৰপিছত আৰু তপন দা অসম মেডিকেল কলেজৰ প্ৰডাক্ট আৰু তপন দা মানে ডাক্তৰটো মই বাদেই দিলো বৰ ভাল খেলুৱৈ
শান্তনু দা আপোনাৰ কথা সকলোৱে কৈ আছে আপুনি কিবা ক'ব নেকি আপোনাক আনমিউট প্লিজ কৰিব পাৰে আৰু মিতালী বাইদেউ আছে আপুনি কিবা ক'ব খোজে যদি আপুনি গৌতম দা বহুতখিনি কথাই ক'লে মই যদিও লিউকেমিয়াৰ লগত ডিৰেক্টলি কানেক্টেড নহয় ডে টু ডে প্ৰেক্টিছত চ' তথাপি আপুনি মানে এইটো কি কয় কাৰ্টি চেলৰ কথা ক'লে চ' আপুনি মানে এইটো কি কাৰ্টি চেল ধৰক মানে এইটো কাৰেণ্ট টাইমত প্লিজ বাইদেউ মিতালী বাইদেউ কওক
একো নাই গৌতম ভাল লাগিলে হেল্ল'
জইন কৰিলে ভাল পালো দে
নাই জইন কৰিলো মই শ্বেয়াৰো কৰিছিলো ঠিক আছে দিয়া
শান্তনু আনমিউট হৈ আছে ন
জানো আচনে নাই নাজানো মই বৰ এইটোৰ লগত বৰ ফেমিলিয়াৰ নহয় কিন্তু গৌতম বৰ ভাল লাগিলে শুনি আৰু আৰু ভালে থাক ভালে হওঁ মইতো সেইবিলাক হেৰি কিন্তু তোৰ কথাখিনি শুনি বা যিখিনি হেৰি লৈ পেলাই মোৰ কাৰণে মই ৰেডিঅ'লজি মানুহ মোৰ অলপ গ্ৰছ হেৰি মাইনুচত ইমান ডিটেইলছত গৈছ কিন্তু মোৰ কাৰণে এটা নতুন অনুভৱ নতুন হেৰি কিন্তু এনেকুৱাবিলাকেই আমাৰ জীৱনত বহুত এটা মানে আপ্লুত কৰে এই কথাবিলাকে ভাল লাগে শুনি তহঁতে এনেকে যে কাম কৰি আছ এই ইমানখিনি নতুন কথা কৈছ এইবিলাক শুনি পেলাই মোৰো এটা নতুন জানিবলৈ শিকিবলৈ এটা উৎসাহ বাঢ়িছে তোৰ কথাখিনি। সেইটোৱে মোৰ কাৰণে এটা ডাঙৰ আশীৰ্বাদ।
Thank you. তুমি ৰিফাত তুমি এটা কিবা সুধিছিলা?
মই একচুৱেলি কাৰ্টি চেলৰ কথা সুধিব খুজিছিলো যে
কোৱা।
আপুনি মানে ব্লাড কেঞ্চাৰত কাৰ্টি চেলৰ ধৰক মানে এইটো কাৰেণ্ট চিটুৱেশ্যনটো কেনেকুৱা, মানে ইজ ইট মোৰ ইন লিম্ফোমাছ অৰ ইজ ইট অলছ' বিং ডেভেলপড ফৰ লিউকেমিয়াছ? So what is the current scene and what is
মানে
its efficacy?
ৰাইট, মানে লিম্ফোমাতে আটাইতকৈ বেছি ডেভেলপড, আৰু আমাৰ মানে লিউকেমিয়াৰ স্পেচত ধৰা একিউট লিম্ফোব্লাষ্টিক লিউকেমিয়াত বেছি ডেভেলপ। তাৰ এটা কাৰণ আছে। কাৰণটো হৈছে মানে এই লিম্ফয়েড কেঞ্চাৰবোৰত ধৰা লিম্ফোমাই হওক বা একিউট লিম্ফোব্লাষ্টিক লিউকেমিয়াত হওক, যিবোৰ চাৰ্ফেচ প্ৰটিন থাকে, সেই চাৰ্ফেচ প্ৰটিনবোৰ মোৰ অৰ লেছ কনষ্টেণ্ট, ৰাইট? মানে পাৰ্টিকুলাৰলি ধৰা কেঞ্চাৰ চেলত যিটো চাৰ্ফেচ প্ৰটিন থাকে মোৰ অৰ লেছ কনষ্টেণ্ট থাকে, গতিকে টাৰ্গেট কৰিবলৈ বেটাৰ। কিন্তু AML-ত হয় কি এই প্লাষ্টিচিটিটোৰ কাৰণে মানে the surface molecules can change, right? That makes it very difficult to target with one CAR-T product. So maybe, conceptually we can think about having 2 or 3 different CAR-T targets, right, to go after AML. Develop হৈ আছে এতিয়া ধৰা, গতিকে মই কৈছো ইণ্ডিয়াটো CAR-T cell particularly ALL acute lymphoblastic leukemia কাৰণে এপ্ৰুভড হৈছে, ভাল কাম হৈ আছে সেইবোৰ ক্ষেত্ৰত। কিন্তু AML-ৰ ক্ষেত্ৰত মানে it's still a challenge, আৰু mostly the challenge is around this situation. The AML cell is much more diverse and much more plastic in that sense.
Okay, thank you. আৰু মানে এটা ধৰক it might be a naive question. So আপুনি মানে এইটো immunotherapy কথা ক'লে যে of course immunotherapy also has its side effects, right? I mean including GI and other. But how about CAR-T cells? Do they have the similar type of side effects or
Yes.
or
Yeah, because again, I mean, as I talked, মানে এইটো মানে accelerator দবাৰ কথা হলতো, কিন্তু accelerator তুমি মানে দবালা কিন্তু হয়তো কন্ট্ৰল নাথাকিবও পাৰে। গতিকে সেই যেতিয়া এই মানে টি চেলবোৰ তুমি যেতিয়া ৰিইনফিউজ কৰিলা পেচেণ্টক, টি চেলটো তুমি পেচেণ্টৰ পৰা ল'লা, লৈ পেলাই মডিফাই কৰি এক্সপেণ্ড কৰি now in millions of numbers you're giving it back to the patient, right? And these cells, once they recognize the cancer cells, they'll start dividing quickly, and when they divide, they release products called cytokines, right? So these cytokines sometimes can create a storm in the patient, and that's sort of the where kind of, and some of these cytokine storms can also have toxicities in the brain, called immune cell related neurologic damages. So that's the critical part. That's where we need to stay vigilant, that how can we, if that happens, how can we dial it down, right? Jodi mane T cell bura, if they're attacking, I mean the expansion and their growth is to a point, or the release of the cytokines is to a point where it's kind of challenging the patient's life, can we dial it down, right? There are different ways of dialing it down. So one of the things is that there's such a smart people, like, so they include something called a suicide gene into the CAR-T cell, right? And if the CAR-T cells are becoming too overactive, right, you can use certain drugs to, because of the presence of the suicide genes, there's only these CAR-T cells will kind of, dial down or die, so that you can kind of stop that, the immune process from hurting the patient.
Thank you. Actually, মই just YouTube-ত এটা প্ৰশ্ন দেখিলো গৌতম দা, which is exactly not related to leukemia, but মই just for the sake of at least responding to our viewer. So প্ৰশ্নটো এনেকুৱা, that is there any role of immunotherapy after enucleation in grade 2 choroidal melanoma as preventive measure?
মানে এইটো ওপৰত অলপ ডিজিজ স্পেচিফিক হ'ল কথাটো
yeah.
ৰাইট আই আণ্ডাৰষ্টেণ্ড কাৰণ কিয় সুধিছে এইটো নহয় কিন্তু হেৰি মোৰ মানে কলিগছ আছে ধৰা মেলানমা ট্ৰিট কৰে পাৰ্টিকুলাৰলি আই বা কাৰণ আইৰ যিটো মেলানমা বহুত সময়ত ব্ৰেইনত যাব পাৰে আৰু মেলানমাৰ ক্ষেত্ৰততো অন্তত ইমিউনথেৰাপিয়ে যথেষ্ট কাম কৰে মানে এক্সাম্পল দিছোঁ ধৰা প্ৰেচিডেণ্ট জিমি কাৰ্টাৰ আছিল হয় নহয় তেখেতৰ মেলানমা একচুৱেলি স্প্ৰেড টু দ্য ব্ৰেইন আৰু তেওঁক ইমিউনথেৰাপি গিভ হিম এবাউট another 10 years of life right literally মানে melanoma disappeared গতিকে melanoma ক্ষেত্ৰত definitely immunotherapy কাম কৰে সেইটো কিন্তু মানে কেৱল মানে ডিজিজ স্পেচিফিক আনসাৰ দিবলৈ মোৰ কাৰণে অসুবিধা কাৰণ এইটো মোৰ ঠিক মানে লাইন নহয় কিন্তু এগেইন ইন মেলানোমা অকুলাৰ মেলানোমা মানে হাইট হোৱা মেলানোমা বা স্কিন মেলানোমা ইমিউনোথেৰাপি যথেষ্ট কাম কৰে
Thank you. So, Gautam, it's like all of switching from the hard topic. So, because our program to like এটা প্ৰধান উদ্দেশ্য হৈছে students in Assam in different fields to inspire them to like progress or get inspired by people like you. আপুনি ধৰক মানে যেহেতু আপুনি ইউএছৰ এটা আগশাৰীৰ কেঞ্চাৰ প্ৰতিষ্ঠানত কাম কৰে MD এণ্ডাৰছন কেঞ্চাৰ চেণ্টাৰত আপোনালোকৰ তাত কেনেকুৱা বা আপোনাৰ বিভাগত বা আপোনালোকৰ ইনষ্টিটিউটত কেনেকুৱা ধৰণৰ ধৰক ট্ৰেইনিং অপৰচুনিটি মানে অসমৰ young at least medico-বিলাকে পাব পাৰে of course it has to be related to people in the field of medicine so কেনেকুৱা ধৰণৰ অপচন থাকিব পাৰে বা what are the things that they can look for maybe somebody in pathology somebody in medicine or oncology কিবা ক'ব পাৰিবনে আপুনি অলপ
rotation definitely arrange কৰিব পাৰি এতিয়া particular ধৰা পেথলজিষ্টে হওক এতিয়া হেৰিৰ পৰা মই এই চণ্ডীগড়ত আছিলো কাৰণ এই চণ্ডীগড়ৰ পৰা আমি কিছুমান ধৰা ইয়াং ফেকাল্টি আহিছে 2-3 মাহ ইয়াত কটাই গৈছে ট্ৰেইনিং লৈ তাৰপিছত হেৰি কি কয় এই BBCI এজন ব্লাড কেঞ্চাৰ স্পেচিয়েলিষ্ট তেওঁ প্ৰায় 3 মাহমান ইয়াত কটাই গৈছে ৱেষ্ট বেংগলৰ পৰাও এজন ব্লাড কেঞ্চাৰ স্পেচিয়েলিষ্ট আহিছে তেওঁ প্ৰায় 3 মাহমান কটাই গৈছে গতিকে ডেট কেন বি এৰেঞ্জড বেলেগ বেলেগ ফৰাম আছে ধৰা মানে কিন্তু সেইটো এৰেঞ্জ কৰিব ডেফিনেটলি পৰা যায় আৰু সেইটো মানে কাৰণ আমাৰ ইনষ্টিটিউটটো মানে ফেকাল্টিটো লাৰ্জলি ইণ্টাৰনেশ্যনেল মেডিকেল গ্ৰেজুৱেট এটা মোৰ ডিপাৰ্টমেণ্টত কৈছো মানে লোকেল ডিপাৰ্টমেণ্টত আমাৰ প্ৰায় 80+% অফ ফেকাল্টি ইণ্টাৰনেশ্যনেল মেডিকেল গ্ৰেজুৱেট গতিকে আমাৰ মানে বেলেগ বেলেগ জেগাৰ লগত আমাৰ কণ্টেক্ট নহয় জানো আৰু আমাৰ গতিকে ট্ৰেইনিং প্ৰগ্ৰামবোৰো আছে আৰু বোলে কিছুমানে বোলে শ্বৰ্ট টাৰ্ম ৰোটেশ্যন ল'ব খোজে ডেট কেন বি এৰেঞ্জড খালি অলপ সময় লাগে কাৰণ বহুত জেগাৰ পৰা বহুত এপ্লিকেণ্ট থাকে গতিকে মানে অলপ সময় লাগে কেতিয়াবা ধৰা 6-8 মাহ 12 মাহ লাগি যাব পাৰে হয়তো এৰেঞ্জ কৰোঁতে কিন্তু এৰেঞ্জ কৰিব ডেফিনেটলি পাৰে
So কোনোবাই যদি ইচ্ছা কৰে ডেফিনেটলি আপোনাক কন্টেক্ট কৰিব পাৰে নহয় জানো
of course yeah
okay thank you আৰু কাৰোবাৰ কিবা প্ৰশ্ন আছে নেকি হিৰেন্দ্ৰ দা আপোনাৰ কিবা আছে নেকি ক'বলগীয়া
গৌতম দা ধন্যবাদ আপুনি ইমান সুন্দৰকৈ কথা কেঞ্চাৰৰ বিষয়ে বিশেষকৈ ব্লাড কেঞ্চাৰৰ কথা ইমান সুন্দৰকৈ ক'লে তপন দাই ঠিকেই কৈছে যে হাতী মাৰি ভুৰুকাত ভৰোৱাৰ নিচিনা কথা ইমান কম সময়ৰ ভিতৰত আৰু ইমানবোৰ বিতংকৈ আপুনি ক'লে মই কিছুমান কথা বুজিলো কিছুমান কথা নুবুজিলো কিন্তু সেইটো চাগে আমাৰ বহু শিক্ষাৰ্থী বা বহু মানুহৰেই হ'ব আপোনাক এটা কথা সুধিব খুজিছোঁ যিহেতু আমাৰ অনুষ্ঠানটোৰ 15 মিনিট শিক্ষাৰ এটা লক্ষ্য যে আমি সাধাৰণ যিসকল নন স্পেচিয়েলিষ্ট থাকে তেওঁলোকক কিছুমান কথা জনোৱাৰ এই কেঞ্চাৰৰ ওপৰত এনেকুৱা এখন কিতাপৰ নাম আপুনি ক'ব নেকি যিখন কিতাপ সাধাৰণ মানুহে পঢ়ি বুজি পায় কেঞ্চাৰৰ কথা আৰু পঢ়ি বুজি পোৱাই নহয় যিসকল এই ৰোগত আক্ৰান্ত হয় তেওঁলোকে হয়তো সাহস পাবও পাৰে তেনেকুৱা এখন কিতাপ কাৰণ আমাৰ এই অনুষ্ঠানটোৰ এটা আমি নতুন পদক্ষেপ ল'বলৈ খুজিছোঁ যিটোত আমি কিছুমান বিশেষকৈ ইংৰাজী ভাষাত প্ৰকাশ হোৱা কিতাপ আমি ভাবিছোঁ যে অসমীয়ালৈ অনুবাদ কৰিলে আমাৰ বহু ক্ষেত্ৰত অকল মানে যিকোনো বিষয়ৰ ফিজিক্সৰ হ'ব পাৰে বা মেডিচিনৰ হ'ব পাৰে ইকনমিক্সৰ হ'ব পাৰে যে আমাৰ ছাত্ৰ ছাত্ৰীসকলৰ লগতে সাধাৰণ ৰাইজৰো উপকাৰ হ'ব গতিকে আপুনি তেনেকুৱা কিবা কিতাপ এখন বা দুখনৰ নাম ক'ব নেকি
হয় এই হেৰি কি মুখাৰ্জী আছিলে
সিদ্ধাৰ্থ মুখাৰ্জী
সিদ্ধাৰ্থ মুখাৰ্জী
Emperor of All Melodies, right? সেইখন মানে that's an amazing book কাৰণ মানে তাত মানে যথেষ্ট মানে নন মেডিকেল মানুহে বুজি পোৱাকে লিখিছে আৰু তাৰ পাছৰ তেওঁ আৰু এখন কিতাপ লিখিছে য'ত তোমাৰ জিন্স নেকি মানে সেই how the হয় নহয় how the genes influence cancer আমি এই মিউটেশনৰ কথা কলো ক্ৰোমোজোমেল এবনৰ্মালিটিৰ কথা কলো এই কথাখিনি যথেষ্ট বুজাব পৰাকে মানে লিখিছে সেই দুখন কিতাপ যদি অসমীয়া অনুবাদ কৰিব পাৰে এইটো খুবেই ভাল হয় ভাল চিন্তা কৰিলা
ইতিমধ্যে অনুবাদ হৈছে
হয় নে
আমাৰ
তপন দা
ফিজিঅলজিৰ যে তৰুণ শইকীয়া ছাৰ আছিলে তেখেতে কৰিছিল অলপ আৰু তাৰপিছত নাৰায়ণ বৰদলৈ দুয়ো মিলি কৰিলে কিতাপখন প্ৰকাশো হ'ল চ' এইখনৰ মই কিছু অংশ পঢ়িলোঁ অনুবাদ কৰা সহজ নহয় মই পঢ়ি পেলাই চাই বুজিলোঁ যথেষ্ট কঠিন আৰু এখন ইমান ডাঙৰ কিতাপ এইখনো আকৌ কোনোবাই সৰুকৈ লিখিব লাগিব তেতিয়াহে মানুহে পঢ়িব এতিয়া তোমাৰ 500-600 পৃষ্ঠাৰ কিতাপ মানুহৰ পঢ়িবৰ ধৈৰ্য নাই 150 পেজৰ ভিতৰত লিখিব লাগিব মানুহে তেতিয়াহে মানুহে পঢ়িব
তেনেকুৱা কিবা কিতাপ আছে নেকি তপন দা নতুনকে ওলাইছে নেকি কেঞ্চাৰৰ ওপৰত
আমি অসমীয়াই লিখিব লাগিব আমাৰ দায়িত্ব আছে
ঠিকেই সেইটো ঠিকেই কৈছে তপন দা আমাৰে দায়িত্ব সেইখন নহয়
দায়িত্ব আমাৰ
অন্যকটো দিব নোৱাৰি এইটো দায়িত্ব
নাই দাদা আপোনালোকে যদি ইংৰাজীটো লিখে আমি অসমীয়ালৈ অনুবাদ কৰাৰ ব্যৱস্থাটো কৰিব পাৰো অসমীয়াত লিখিলেতো ভালেই পোনপটীয়াকে
হয় বৰ ভাল কথা
দাদা আপুনি সামৰণি মাৰিব পাৰে অনুষ্ঠানৰ আজি
গৌতম দা পুনৰ ক'লো আপুনি ইমান সুন্দৰকৈ আমাক কেঞ্চাৰৰ বিষয়ে বিশেষকৈ তেজৰ কেঞ্চাৰ কৰ্কট ৰোগৰ বিষয়ে আমাক জনালে আপুনি আপোনাৰ সময় আমাক দিয়াৰ কাৰণে 15 মিনিট শিক্ষাৰ তৰফৰ পৰা অশেষ ধন্যবাদ জ্ঞাপন কৰিছোঁ আশা ৰাখিছোঁ আপোনাৰ কথাখিনিৰ জৰিয়তে এই অনুষ্ঠানত উপস্থিত থকা আমাৰ তিনিওটা প্লেটফৰ্মত উপস্থিত থকা শ্ৰোতা দৰ্শকসকল নিশ্চয়কৈ উপকৃত হৈছে আৰু ভৱিষ্যতে যিসকলে আমাৰ ৰেকৰ্ডেড ভিডিঅ'টো চাব তেওঁলোকো উপকৃত হ'ব তাতোকৈ ডাঙৰ কথা যে যিসকল ৰোগী এই ৰোগত আক্ৰান্ত হয় তেওঁলোকৰ কাৰণে কেঞ্চাৰ কৰ্কট ৰোগ এটা অভিশাপ ৰূপে আহে তেওঁলোকৰ পৰিয়ালৰ কাৰণে এইটো এটা খুব ষ্ট্ৰেছফুল অভিজ্ঞতা হয় আৰু তেনে অৱস্থাত তেনে পৰিস্থিতিত তেওঁলোকে যেতিয়া জানে যে আমাৰ মাজৰে একোজন এনেকুৱা বিশেষজ্ঞ আছে যিয়ে এই ৰোগটোৰ বিষয়ে খুব বিতংকৈ জানে আৰু তেওঁলোকে চিকিৎসা কৰে সেইটো আমাৰ বোধেৰে এটা ডাঙৰ কথা এটা ডাঙৰ সাহস আপুনি থকা তপন দা থকা সংগীতা থকা এইবিলাক আচলতে আমাৰ কাৰণে আৰু তেওঁলোকে জনাটো যে আপোনালোক আছে আপোনালোকে এই বিষয়টোৰ ওপৰত অধ্যয়ন কৰি আছে চিকিৎসা কৰে সেই কথা জনাটো খুব দৰকাৰী প্ৰয়োজনীয় বুলি আমি ভাবোঁ আশা কৰোঁ আপোনাৰ এই বক্তৃতাটোৰ জৰিয়তে তেওঁলোকে সেই সাহসকণ পাব কথাখিনি আপুনি বুজোৱা কথা আটাইখিনি বুজি নাপালেও সেই সাহসটো পোৱাটো এটা ডাঙৰ কথা তাৰ কাৰণে আপোনাক অশেষ ধন্যবাদ ইয়াত উপস্থিত থকা আটাইলৈকে আমাৰ 15 মিনিট শিক্ষাৰ তৰফৰ পৰা কৃতজ্ঞতা জ্ঞাপন কৰিছোঁ আশা ৰাখিছোঁ যে আপোনালোকে আমাৰ অনুষ্ঠানবোৰ চাই থাকিব আৰু যিসকল উঠি অহা শিক্ষাৰ্থী অসমত আছে অসমৰ গাঁৱে ভূঞে চুকে কোণে তেওঁলোকক আমাৰ এই অনুষ্ঠানটোৰ বিষয়ে জনাব আমাৰ কাৰণে ভাল কথা যে 2025ৰ শেষৰ ফালে আমি এই অনুষ্ঠানবোৰৰ ভিত্তিত ৰচিত এখন কিতাপ পাখি মেলা সপোন আমি গুৱাহাটী গ্ৰন্থমেলাত আনুষ্ঠানিকভাৱে উন্মোচন কৰিছোঁ তাত আমাৰ এই অনুষ্ঠানত প্ৰকাশ পোৱা প্ৰচাৰিত বিভিন্ন অনুষ্ঠানত যিবিলাক অনুপ্ৰেৰণামূলক কাহিনী আছে, আমাৰ ভিতৰুৱা জেগাৰ পৰা ওলাই আহি পৃথিৱীৰ বিভিন্ন ঠাইত যিসকলে সফলতা লাভ কৰিছে তেওঁলোকৰ কাহিনীৰ ভিত্তিত মুনীন্দ্ৰ বৈশ্যই যিখন কিতাপ লিখি উলিয়াইছে সেইখন ইতিমধ্যে প্ৰকাশ কৰিছোঁ। আমাৰ আৰু এনেকুৱা কিছুমান কাৰ্যসূচী আগলৈ আমি কৰিম বুলি ভাবিছোঁ। আশা ৰাখিছোঁ আমি সকলোৰে সহযোগিতা লাভ কৰিম। পুনৰবাৰ আপোনালোক আটাইকে ধন্যবাদ জনাই, গৌতম দা আপোনাক ধন্যবাদ জনাই আমাৰ অনুষ্ঠানৰ সামৰণি মাৰিছোঁ। পৰৱৰ্তী অনুষ্ঠান ফেব্ৰুৱাৰীৰ 8 তাৰিখে, সেই অনুষ্ঠানত মই 2025 চনৰ অৰ্থনীতি বিজ্ঞানৰ নোবেল বঁটাৰ বিষয়ে কিছু কথা আলোচনা কৰিম। ধন্যবাদ। আৰু লগতে আপোনালোকক এইটো কথাও জনাই যাওঁ যে ফেব্ৰুৱাৰীৰ 22 তাৰিখে আমাৰ যিটো পঞ্চম প্ৰতিষ্ঠা দিৱস বক্তৃতা, সেই বক্তৃতাটো এই প্লেটফৰ্মত অনুষ্ঠিত হ'ব আৰু বক্তৃতা দিব ডক্টৰ অনুপম দত্তই। ধন্যবাদ।
অলপ দেৰি ৰ'ব পাৰে। ধন্যবাদ। অকণ দেৰি থাকিব পাৰে গৌতম দা।
নহয় মানে
লাইভটো শেষ হ'ল।